Hello everyone, welcome back to my YouTube channel, Life Sciences by Tumi Silani. This is a YouTube online teaching platform where I give full-length lessons on different topics of the grade 12 curriculum. Soon and very soon, I will be posting content on grade 11 curriculum. So please bear with me if you are a grade 11 watcher or you are a teacher also teaching grade 11. Okay. Please do like, comment, and subscribe. Share the link to this video to your fellow learners or to your fellow colleagues if you are a teacher. And let's get right into it. My name is Tumi Silani. I am a life sciences teacher at Milner High School in the Northwest Province. This will be lesson 2 on our lesson series on the topic meiosis. We will be looking at the process of meiosis 1 as well as meiosis 2. On the previous lesson 1, we looked at very important introductory concepts for you to be able to form a background knowledge on today's lesson and the lessons going forward. So before we delve into the lesson, let us look into how the lesson series is divided. I have decided to do the topic of meiosis in 4 distinct lessons that are contained in 4 individual videos. Okay, let us break down the lessons. In lesson 1, we looked at the introduction to meiosis. We looked at what a diploid cell is versus what a haploid cell is. We looked at somatic cells and how they differ from gametes. We looked at the human life cycle, the chromosome structure, as well as the karyotype. There was a long lesson, but it was necessary for us to understand these preliminary concepts before we delve into the other concepts of meiosis, or rather the other lessons of meiosis. Today, it's lesson 2. We will be looking at the process of meiosis 1 and meiosis 2. On a separate video, on the next lesson, lesson 3, we will be looking at genetic variation, how meiosis contributes towards genetic variation. It does that in 2 ways. We will be looking at crossing over as well as random arrangement of chromosomes. The 4th and final lesson, we will be looking at abnormal meiosis. And under abnormal meiosis, it is prescribed for you to only know about non-disjunction. Okay, without wasting any more of your time, let us go straight into the lesson. Meiosis, the process. Before we look into meiosis 1 and meiosis 2, here is a very quick overview of the process of meiosis. Okay, now, firstly, interphase will take place. Remember guys, interphase is not part of meiosis. Interphase happens before meiosis. Okay, it is not part of meiosis, it happens before meiosis. Remember, we said interphase is a phase, a period of time where the cell prepares to divide. So what are the things that happen during interphase in preparation for the cell to divide? Very, very quickly, I am not going to be repeating the things that are occurring in meiosis in greater detail. If you need details about this, please go to lesson 1 to revise, to check what happens during interphase. So during interphase, DNA replication will take place. There will be a doubling of the cell's DNA or genetic material. All unreplicated chromosomes will now be replicated. So there is a cell before interphase. As you can see, if it was a human cell, it would contain 46 chromosomes. Now, in this particular instance, our cell contains 4 chromosomes. For the sake of teaching, for the sake of convenience, for the sake of space, this is a diagram that only contains 4 chromosomes. So going forward, we will be looking at a diagram containing 4 chromosomes. However, if it was a human cell, it will be containing 46 chromosomes. So these 4 chromosomes represent our diploid number, our 2 sets of chromosomes. As you can see, there are different colors. Therefore, it is 2 sets of chromosomes. One set of chromosomes is red and one set of chromosomes is blue. Okay, so yes. So when this cell prepares to divide, DNA replication will take place, and these unreplicated chromosomes will now be replicated, as you can see over there. So these red chromosomes, there they are now replicated. They contain 2 chromatids each, and the blue chromosomes, the blue unreplicated chromosomes, after interphase, will now be having 2 chromatids each. Okay, now when the cell is like this, containing replicated chromosomes, it is now ready to divide. So it will undergo meiosis 1. Meiosis 1 is divided into 4 phases. That will be prophase 1, metaphase 1, anaphase 1, as well as telophase 1. Now, immediately after telophase 1, there will be no interphase again. Interphase just happens once before meiosis starts, before meiosis 1. So after meiosis 1, there is no telophase. There is no interphase again. Okay? So after meiosis 1, we will be having 2 daughter cells, 1, 2, each containing half the number of chromosomes from the original mother cell. So the mother cells, we started with 4 chromosomes, that is the diploid number. And then after meiosis 1, we will divide the number of chromosomes into 2 each. If this was a human cell, it would be 23 chromosomes in this cell and 23 chromosomes in that cell. So immediately after telophase 1, we are going to go through to prophase 2, then metaphase 2, anaphase 2, as well as telophase 2. Now, after telophase 2, what is going to happen? Each daughter cell will divide to form 2 daughter cells each. Okay, so each daughter cell divides to form 2 additional daughter cells each. So at the end of telophase 2, that is the end of meiosis 2, and that is the end of meiosis period. Okay, so at the beginning of meiosis 1, we start with 2 daughter cells and we finish with 4 daughter cells. And as you can see, there is no halving of chromosome numbers during meiosis 2. We only reduce the number of chromosomes from diploid to haploid in meiosis 1. In meiosis 2, we still have 2 chromosomes in each daughter cell, but the difference between the chromosomes in the daughter cells of meiosis 1 and the chromosomes in the daughter cells of meiosis 2 is that in meiosis 1, the chromosomes are replicated, 2 replicated chromosomes, but then at the end of meiosis 2, we still have 2 chromosomes, but these 2 chromosomes are unreplicated. Okay, so if you are confused at any way, it's okay, it's fine. This was just an overview. None of this is going to make sense now. Okay, so only after we have dealt with the process of meiosis 1 and meiosis 2 will this make sense to you. Okay, now the first phase of meiosis is prophase 1. Let us look into that. Okay, guys, like we said, meiosis has two cell divisions, meiosis 1 as well as meiosis 2. Now, for the sake of convenience, please use the abbreviation PMAT. We know that the phases of cell division, both meiosis and mitosis, are prophase, metaphase, anaphase, as well as telophase. Okay? PMAT. P prophase, M metaphase, A anaphase, T telophase. However, because meiosis has meiosis 1, and then the daughter cells of meiosis 1 will divide into meiosis 2, we have two meiotic divisions. Okay? The cell divides twice. Now, we need to attach a 1 after every phase of meiosis 1. Why do we need to attach a 1 after every phase of meiosis? Is because meiosis has two divisions. Okay? If you just write prophase, metaphase, anaphase, telophase, when you are referring to the phases of meiosis, you will be discredited. You will be discredited because the marker will assume that you are referring to phases in mitosis, since mitosis only has one cell division. You don't need to attach 1 or 2 in mitosis. However, because meiosis contains two divisions, meiosis 1 and meiosis 2, when you are referring to a particular phase in meiosis, you need to attach 1 or a 2 after the phase. Now, because you are used to just writing prophase, metaphase, anaphase, and telophase from grade 10, this will take some time to get used to. But guys, please do not forget to attach a 1 or a 2 when you are referring to phases of meiosis. Okay. Okay, now the first phase of meiosis 1 is called prophase 1. Remember, we said that we use the abbreviation P. We use the abbreviation PMAT. This shows the order in which the phases follow. Okay, so prophase, metaphase, anaphase, telophase, and because we are dealing with meiosis 1, it will be PMAT 1, meaning prophase 1, then followed by metaphase 1, then followed by anaphase 1, then followed by telophase 1. This is not an official abbreviation for the phases, guys. This is not an official abbreviation. This is only a study technique that will help you memorize the phases of meiosis and the order in which they appear. So because it is PMAT, the first phase of meiosis is prophase 1. So we start with the cell being like this, a normal cell. We will find replicated chromosomes inside the nucleus. Let us now label what the different parts are. So our cell would contain a membrane on the outside, being the cell membrane, and the void space inside that cavity. That space within the cell will be filled by a jelly-like or a semi-liquid called the cytoplasm. And there we have the nucleus, and then the nucleus we know that is bound by or surrounded by a nuclear membrane. Inside the cell, when the cell is dividing, remember we said that the DNA will condense to form distinct chromosomes. So that in particular is a replicated chromosome. And then those two parts form the nucleus. And then just outside the nucleus, you will see in your exam question papers, usually there is a pigmented dot just outside the nucleus, and that is known as the centrosome 1. centrosome. Only one centrosome. Not to be confused with the centromere. Okay? Not to be confused with the centromere. The centrosome is a structure that is found outside of the nucleus, and the centromere, remember, is in the center of the replicated chromosome. It is a structure that holds two chromatids together. Okay, so centromere is in the center of a chromosome, and then the centrosome is outside the nucleus. Okay, guys? Now remember, in terms of the phases of meiosis, your job is to be able to study theoretically and be able to tell what happens to the cell in each phase of meiosis. What happens to the cell and the chromosomes in each phase of meiosis. So you must be able to memorize the occurrences that occur bullet by bullet during each phase of meiosis. Okay, so now in prophase 1, you must be able to describe particularly how the cell behaves during prophase 1, metaphase 1, anaphase 1, telophase 1. Same goes with the phases in meiosis 2. Okay, now we will be learning theoretically, and I will be giving you visuals as to what happens in each phase. But it is your job. It is your job to be able to memorize what happens to the cell during each phase of meiosis. Because in the exam, you might be asked to describe what happens in a particular phase of meiosis, or you might be given a diagram showing occurrences that are happening in a particular phase. You may be asked which phase is shown here, and after that, of course, you will be asked why do you say that is the particular phase that you have identified. Okay, so let's check what happens during prophase 1. During prophase 1, the nuclear membrane will disappear. So there we go, our nuclear membrane has disappeared. So now the chromosomes are just floating inside the cytoplasm. So the nuclear membrane disappears. Replicated chromosomes come together to form homologous pairs. So originally, they would just be floating around the nucleus, but these replicated chromosomes will come together to form homologous pairs. There we go. Now they are in homologous pairs. How can we tell that they are in homologous pairs? We can tell because they are, from this diagram particularly, we can tell because they are the same size. Okay, the next thing that happens is that crossing over will take place. Guys, I am not going to be talking about crossing over in this lesson. I will talk about crossing over in lesson 3 when we deal with genetic variation. So I will specifically talk about crossing over in that lesson. So for now, just know that crossing over is taking place. How do you know that crossing over is taking place? As you can see, the chromatids are overlapping. Okay, the chromatids of homologous chromosomes are overlapping. Don't worry about crossing over for now. You will understand crossing over particularly when we look into lesson 3. Okay, another thing that happens is that this guy over here, this structure, the centrosome, splits and it forms centrioles, and then the centrioles will start moving to the opposite poles of the cell. Now we have 3 words here that sound alike and that confuses many learners. Centromere, remember, centromere is at the center. Centromere is at the center of the chromosomes. It is holding the 2 chromatids together. And then we have centriole, singular, okay? Sorry, see, I'm already confused myself. Centrosome, guys, centrosome, singular, just one centrosome, one centrosome. And the way to memorize the centrioles is that the centrioles move to the poles of the cell. Centrioles for poles, okay? They rhyme. That was the end of prophase 1. So metaphase 1, we end up where we continue from where we left off. I was just explaining to you to memorize centriole. Centrioles are at the poles of the cell, okay? And then centromere is at the center of the chromosome, okay? So the centrosome had split and the centrioles now have moved to the poles of the cell. So there we have our centrioles at the poles of the cell. Okay, now metaphase is a very short and sweet phase of meiosis. It has only 2 steps that you need to know for grade 12 purposes. Chromosomes align randomly at the equator of the cell in homologous pairs. Chromosomes align randomly in the equator of the cell in homologous pairs. Now, what is the equator? The word equator just means the middle of the cell. So the word equator just means middle of the cell. So it can be the vertical middle of the cell like that, or it can be the horizontal middle of the cell like that. So chromosomes align randomly at the equator of the cell in homologous pairs, guys. Examiners are very particular for you to mention certain phrases in your answers. They line themselves at the equator of the cell in homologous pairs. The second thing that happens is that spindle fibers attach to the centromeres of the homologous chromosomes. Now, what are spindle fibers? Spindle fibers are threads, okay? These threads are produced by the centrioles, and they attach themselves to the centromeres of the homologous chromosomes. Now, check what is happening. There is our spindle fibers. I'm not sure if you can see them. There is a spindle fiber. There's another spindle fiber. There's another spindle fiber. There's another spindle fiber. Look at what they are doing. They are moving and they are attaching themselves to the centromeres of the homologous chromosomes. Okay, so there are 2 things that happen during metaphase. Chromosomes align themselves randomly to the equator of the cell in homologous pairs, and spindle fibers attach to the centromeres of the homologous chromosomes. That's all that you need to know. Okay, let us move on to our next phase. Anaphase 1. We start as the cell being like this. Okay, remember from the previous phase, the homologous chromosomes or chromosomes aligned themselves at the equator of the cell randomly, and then secondly, spindle fibers formed and they attached themselves to the centromeres of the chromosomes. Now, after that, what happens? So from the beginning of anaphase 1, the spindle fibers are going to contract. Now, the word contract sounds difficult, but it just means that these spindle fibers pull. So contract just means pulling. Okay? So it means this spindle fiber is going to pull to that direction. This spindle fiber will pull to that direction. No, sorry, wrong direction. That spindle fiber will pull to that direction. That spindle fiber will pull towards that direction. And this spindle fiber will pull towards that direction. Okay, so when the spindle fibers contract, or when they pull the chromosomes will move to the opposite cells of the poles. Okay, so the spindle fibers contract whole chromosomes, guys, important, whole chromosomes are pulled to the opposite poles of the cell. Whole chromosomes are pulled to the opposite poles of the cell. So now let's check what happens. Look at what happens. As the spindle fibers are contracting, whole chromosomes are pulled to the opposite poles of the cell. So they are moving from the equator of the cell to the opposite poles of the cell. All right? As you can see, we have half the number of chromosomes on the left pole and half of the number of chromosomes on the right pole. If this was the equator of the cell, this one cell, half the chromosomes are to the left pole, half the chromosomes are to the right pole. They were pulled to the opposite poles of the cell due to the contracting spindle fibers. All right, so if this was a human cell, we would have 23 chromosomes on one pole of the cell and 23 chromosomes on the other pole of the cell. And that is the end of anaphase 1. The last phase of anaphase 1, of meiosis 1, we end up having half the number of chromosomes on the left pole and half the number of chromosomes on the right pole. That's where we ended with anaphase 1. So here, what's going to happen? Each pole of the cell contains half the number of chromosomes. That is correct. Nuclear membranes will reform around each set of chromosomes. Let's see if that happens. There's the nuclear membrane reforming on this set of chromosomes on the left pole. And we have another nuclear membrane reforming on this set of chromosomes on the right pole. And then cytokinesis will occur. Cytokinesis, kinesis means division, cyto of the cytoplasm. So at this point, it is still one cell. Why? Because it shares a cytoplasm. It will only become two cells if it does not share a cytoplasm. So when cytokinesis happens, cytokinesis will divide the cytoplasm in half. And only then does that one cell become two cells. So cytokinesis occurs, which results in two haploid daughter cells that are genetically different from each other. Okay, so let's see what happens. So cytokinesis has occurred. There we go, the cytoplasm has divided. The word cytokinesis, like I said, means division of the cytoplasm, which will result in what? We have two daughter cells. One cell, daughter cell 1, the other cell, daughter cell 2. Okay? And then, what more do we need to say about these two daughter cells? So, two daughter cells that are genetically different from each other. When you look at the chromosomes, each chromosome has a different combination of colors. Each color represents a combination of genes. So these daughter cells are different from each other. Okay? And they are also haploid. Remember, at the beginning of meiosis 1, we started with having 4 chromosomes in the cell. Right? Now, after meiosis 1, each daughter cell contains how many? 2 chromosomes. If it was a human cell, we would have started with 46 chromosomes, the diploid number, which after meiosis will result in half of that, which is going to be 23 in each one of the 2 daughter cells. Okay. Moving along. Overview after meiosis 1. We started off with a mother cell with 4 chromosomes. Remember, the different colors show the combination of genes. We will look into why they become genetically different when we look at genetic variation on a separate note. For now, we started off with 1 cell which had a diploid. Remember, whatever number we begin with is diploid. And then whatever number we end with after meiosis 1 and meiosis 2 will be haploid. So we started off with a mother cell containing a diploid number of chromosomes of 4. After meiosis 1, we reduced the number of chromosomes into 2 chromosomes each. Okay? And another thing that we must note, when we look at every single chromosome in the daughter cells, they are genetically different from each other. They have different combination of genes. Another thing that we must note is that the chromosomes are still replicated. They still have 2 chromatids each. Okay, let's articulate that in a sentence. After meiosis 1 starts with prophase 1, metaphase 1, anaphase 1, and telophase 1. There is no interphase again after meiosis 1. No interphase again. We move directly to meiosis 2. Now, from here on, we are going to be looking at meiosis 2. Remember, our abbreviation is PMAT. So we started with PMAT 1, meaning prophase 1, metaphase 1, anaphase 1, as well as telophase 1. Now, there is no interphase between meiosis 1 and meiosis 2. We finish with telophase 1. We go directly into prophase 2. So PMAT 2 will be prophase 2, metaphase 2, anaphase 2, as well as telophase 2. Just a reminder, your job is to be able to memorize and describe the different occurrences that happen within the cell in each phase. So learn it theoretically by memorizing the steps and visually to be able to identify a phase, and immediately you must be able to tell what is happening within the cell, and what is happening within the cell will inform what phase that cell is going through in terms of meiosis. Okay, so let's look into meiosis 2. Right, so this is where we ended off. 2 daughter cells that are genetically different from each other. They have a haploid number of chromosomes. Okay, so after telophase 2, this is where we ended off. We go directly to prophase 2. After telophase 1, this is where we ended off. We go directly to prophase 2. Now in prophase 2, what happens? The nuclear membrane disappears again in both cells. Chromosomes are arranged individually in the cell. They are not arranged in homologous pairs. They are arranged individually. Okay? And then crossing over does not take place. Remember, crossing over takes place between homologous chromosomes. So since these chromosomes are arranged individually, there is no crossing over. And then the centrosome splits and forms centrioles, which move to opposite poles of the cell, just like in prophase 1. So there is our centrosome splitting into centrioles, and the centrioles are moving to opposite poles of the cell. Right, so that's the end of prophase 2. From there, okay, now in metaphase 2, we start with where we left off with prophase 2. Okay, guys, this chromosomes are individual. As you can see, they are not in homologous pairs. There are different sizes in each cell, therefore they are not homologous. They are arranged individually. And as you can see, we no longer have the nuclear membrane. It has disappeared. And another thing, the centrosome has split to form centrioles, which are at the poles. Okay, guys? So, what happens? Chromosomes align individually and randomly at the equator of the cell. Okay? During metaphase 1, remember, they would be arranging in homologous pairs, but in metaphase 2, they are arranging individually at the equator of the cell. Now let's check if that is going to happen. There we go. Remember, we said that the equator just means the middle of the cell. It can be a vertical middle or it can be a horizontal middle. Okay, so chromosomes align individually and randomly at the equator of the cell. The second thing that happens is that spindle fibers attach to the centromere of the individual chromosomes. Remember, guys, the job of the centrioles is to produce spindle fibers. So let's check the spindle fibers. They are forming and attaching themselves to the centromeres of the individual chromosomes. In the second cell, they are going to form and attach themselves to the centromeres of the chromosomes. And just like that, metaphase 2 is over and done with. Right, now in anaphase 2, we continue from where we left off, okay? In anaphase 2 is similar to anaphase 1, but there are some key differences. Spindle fibers contract just like in anaphase 1, and the chromosomes now will split. As you can see there. So the chromosomes have split in half. When the spindle fibers contract, the chromosomes will split in half, like that. Okay? And when the chromosomes split, chromatids or unreplicated chromosomes are pulled to the opposite poles of the cell. Remember, in anaphase 1, we said whole chromosomes are pulled to the opposite poles of the cell. But here, the language now changes. We say chromatids. Remember that the chromosomes had split in half, so the chromatids will move to the opposite poles of the cell, or you can use the word unreplicated chromosomes. Move to the opposite poles of the cell. Okay? Now, this is where we are at the moment. As you can see, each cell there we go. Each cell has 2 chromatids, each at the opposite poles of the cell. All right? Now, the language now changes. In anaphase 2, we said that chromatids are pulled to opposite poles of the cell as the spindle fibers contract. We said chromatids, or unreplicated chromosomes. But now here in telophase 2, these chromatids, we do not refer to them as chromatids. Each individual structure, this one and this one, that one and that one, we say they are chromosomes. What's the difference between this one and the chromosome we had before? These chromosomes each contain one chromatid. Therefore, they are unreplicated chromosomes. They are unreplicated chromosomes. Okay? So what happens during telophase 2 is that the nuclear membranes reform around each set of chromosomes. Let's see if that's what happens. So there we have nuclear membranes forming around each set of chromosomes. Next, cytokinesis occurs. Remember, cytokinesis means the division of the cytoplasm. And then, after cytokinesis, we are going to have 4 daughter cells. 4 daughter cells that are genetically different from each other. There we go. We now have 1, 2, 3, 4 daughter cells. Okay? When you look at the chromosomes, remember the colors of the chromosomes represent combination of genes. No chromosome is the same as in another cell. So each cell has different combination of genes. Therefore, the cells are genetically different from each other. All right? Another thing that we must note is that how many chromosomes do we have in this cell? We have 1 and we have 2 chromosomes, not 2 chromatids, 2 chromosomes. These chromosomes are unreplicated. This cell also has 2 chromosomes. That cell also contains 2 chromosomes. That cell also contains 2 chromosomes as well. Okay. Okay guys, now here I have a video simulation showing the steps of meiosis 1 as well as meiosis 2. I will be sticking to the descriptions that we have talked about in the previous slides. So each phase I will be describing how the cell is behaving in accordance to the descriptions of the different phases of meiosis that we have discussed in the previous slides. Okay. So we start off by having the DNA there is condensing to form replicated chromosomes. So there we have our replicated chromosomes, and then the 2 chromatids are joined together by a centromere. Number 1, we said that during prophase 1, replicated chromosomes come together to form homologous pairs, and crossing over now is taking place. Guys, we said that I will be speaking about crossing over in a separate video in lesson 3. Okay, I will be talking about crossing over in lesson 3. So the video now is showing the process of crossing over. Don't worry about crossing over now. We will look at that's at a separate video. As you can see over there, what's going to happen next? So firstly, homologous chromosomes came together to form homologous pairs. Crossing over has taken place. Okay. The nuclear membrane, as you can see over there, will start to disappear. what do we see at the top? The centrosome has split and the centrioles are moving to the opposite poles of the cell. So there, the centrioles are moving to the opposite poles of the cell, and then the nuclear membrane has disappeared. So this is the beginning of meiosis of metaphase 1. During metaphase 1, what did we say is going to happen? We said that spindle fibers, we said that the chromosomes will align themselves at the equator in homologous pairs. So they have lined themselves at the equator in homologous pairs, and then the spindle fibers, they have attached themselves to the centromere of the homologous chromosomes. Continuing. So this is now the beginning of anaphase 1. During anaphase 1, during anaphase 1, what do we expect to happen? We expect that the spindle fibers will contract. When they contract, whole chromosomes will be pulled, guys, not pushed, pulled to the opposite poles of the cell. Let's see if that's going to happen. So the spindle fibers are contracting and whole chromosomes, as you can see over there, are being pulled to the opposite poles of the cell. That's what happens during anaphase 1. So we're going to end up with a situation where each pole of the cell is going to contain half the number of chromosomes. Let us check. So this pole of the cell will contain 2 chromosomes. That pole of the cell will contain 2 chromosomes. If this was the human cell, we would have 23 chromosomes on the left pole and 23 chromosomes on the right pole. So now when we have half the number of chromosomes in each pole of the cell, that would signal the beginning of telophase 1. Now, during telophase 1, as you can see, we have half the number of chromosomes at the opposite poles of the cell. Cytokinesis is going to happen. What did we say cytokinesis is? We said that cytokinesis is the division of the cytoplasm. Remember, the word kinesis means to divide. Cyto refers to the cytoplasm. So cytokinesis is the division of the cytoplasm. Now, after cytokinesis has occurred, the cell now has divided. We have 2 daughter cells. We have 2 daughter cells, and these 2 daughter cells contain half the number of chromosomes compared to what we started with. All right, we started with 4, so each daughter cell now will contain 2 chromosomes each, a haploid number of chromosomes. So by that, we have reduced the number of chromosomes in each cell from diploid to haploid. Okay, guys? Now, the chromosomes are still replicated. The chromosomes are still replicated. They contain 2 chromatids each. And the cells are genetically different from each other because they contain a different combination of chromosomes with a different combination of genes. Okay, guys? So after this, we said that there is no interphase again. We are going straight to PMAT2, meaning after telophase 1, we are going straight to prophase 2, metaphase 2, anaphase 2, as well as telophase 2. So what happens on the left cell will also happen on the right cell. Let us continue the video. All right, now this is meiosis 2. Like I said, each cell is supposed to divide to form 2 additional cells. So what is happening there? We expect that the nuclear membranes on both cells will disappear. The centrosome, as you can see, has already split, okay, to form centrioles, and the centrioles will move to the opposite poles of the cell. Let's check if that is what's happening. That is precisely what is happening. Something that I want you to notice here, is that the chromosomes, the replicated chromosomes, now are arranged individually. They are not arranged in homologous pairs. Remember, homologous pairs, the first thing that tells us that chromosomes are homologous is the fact that they are the same size. When you check those chromosomes, they are not the same size. Okay, guys? So, the chromosomes are arranged individually. So that is the end of prophase 2. And then now we are beginning metaphase 2. The individual chromosomes are lining to the equator of the cell. Spindle fibers are attaching to the centromeres of the individual chromosomes. Okay, so when you check here, we have the spindle fiber attaching to that side of the chromosome, and the spindle fiber on the other side is attaching to the other side of the chromosome. Okay, so metaphase 2, individual chromosomes line in the equator of the cell, and the spindle fibers attach to the centromeres of the individual chromosomes. During anaphase 2 now, what do we expect to happen? The spindle fibers are going to contract. When the spindle fibers contract, check what is happening. The chromosome is split in half. Okay? The chromosome is split in half, and chromatids, or you can say unreplicated chromosomes. Remember, unreplicated chromosomes are chromosomes with one chromatid. Okay, so chromatids or unreplicated chromatids, after the replicated chromosome splits, the unreplicated chromosomes now will move to the opposite poles of the cell. Emphasis, chromatids or unreplicated chromosomes will move to the opposite poles of the cell. And that is what precisely is happening. So there are the chromatids or unreplicated chromosomes moving to the opposite poles of the cell. That is the end of anaphase 1. And during telophase 2, cytokinesis is going to happen. And the two cells will divide to form two more cells. Okay? Let us check if that is going to happen. Yes, there, cytokinesis is happening. So we are going to end up having how many daughter cells? 4 daughter cells. I want us just to go back a bit. Okay, now we are at the end of meiosis 2. Let's go back to the overview again. We started off with having one original cell that we call the mother cell. As you can see over there, we started off with the cell having unreplicated chromosomes. And then interphase happened. Remember, guys, interphase happens before cell division. It is a phase or a period of time where the cell prepares to divide. The most important thing that happens during interphase is DNA replication. The DNA contained in each unreplicated chromosome will make an identical copy of itself. Okay? So the unreplicated chromosomes containing one chromatid, these chromatids will be doubled, meaning the DNA within the chromatids of the unreplicated chromosome will make one additional copy. Okay, and then doubling of DNA or genetic material occurs during interphase. Unreplicated chromosomes will become replicated, as you can see over there. So when the what is this? When the chromosomes are replicated, meaning containing two chromatids, now the cell is ready to divide. As you can see, the red chromosomes, unreplicated chromosomes, they were unreplicated. They contained one chromatid each, and then a copy of that chromatid was made. Now they are going to have two chromatids. Same goes with the blue chromatids. A copy of this chromatid was made, and then the blue chromosomes now have an additional chromatid, and now they have two chromatids each. That is a result of DNA replication, which happens during interphase. Okay, now when the cell contains a diploid number of chromosomes and those chromosomes are replicated, the cell now is ready to divide. So after interphase, the cell goes into meiosis 1. Remember, meiosis 1 is P-MET1. Prophase 1, metaphase 1, anaphase 1, as well as telophase 1. Okay, so after meiosis 1, we are dividing a diploid mother cell, meaning this cell is diploid, so it is a somatic cell. Remember, we said that somatic cells are always diploid. They always contain two sets of chromosomes. Okay? And this diploid cell will be divided into two haploid daughter cells. There we go. They are haploid. Why? Because they contain half the number of chromosomes from the mother cell that we started with. Okay, guys? And then another thing to note is that, okay, on top of them being haploid, they contain a different combination of chromosomes with a different combination of genes. Therefore, they are genetically different from each other because of crossing over that took place during prophase 1. Hence, they have different colors. Okay, the different colors show the different combination of genetic material that is found on the chromosomes itself. The different combination of genetic material or the different combination of DNA. Okay, guys? Another thing, one last thing that we need to note about the daughter cells after meiosis 1 is that the chromosomes are still replicated. All right. So each daughter cell is going to divide by meiosis 2, like we saw in the lesson. So after meiosis 2, PMET2, prophase 2, anaphase 2, prophase 2, metaphase 2, anaphase 2, as well as telophase 2, each daughter cell from meiosis 1 is going to divide. So this cell is going to divide, and that cell is also going to divide. We are going to end up having 4 haploid daughter cells, as you can see over there. Okay, and the chromosome number is not halved during meiosis 2. Guys, very important. The halving of the chromosome number only occurs during meiosis 1. During meiosis 2, we do not halve the number of chromosomes again. Okay, so for instance, we started off, we started off with a diploid cell containing 4 chromosomes at the beginning of meiosis 1. And then we have 2 daughter cells that contain half the number of chromosomes, which is a haploid number of 2. Okay? So, during meiosis 2, we start by dividing daughter cells with 2 chromosomes each, And then at the end of meiosis 2, we are going to end with exactly that number that we started with at the beginning of meiosis 2, okay, which is 2. The only difference between chromosomes within each daughter cell at the end of meiosis 2 compared to chromosomes in each daughter cell at the end of meiosis 1 is that the chromosomes at the end of meiosis 1 will be replicated. However, the chromosomes at the end of meiosis 2 will be unreplicated. Okay, so that's what we see over there. So at the end of meiosis 1, we have 2 chromosomes. And then at the end of meiosis 2, we also have 2 chromosomes. But these chromosomes are unreplicated. They are unreplicated chromosomes. They are not just chromatids. They are unreplicated chromosomes. Now, if you can check, look at this cell and look at that cell over there, the original mother cell. So the original mother cell, as you can see, also contained unreplicated chromosomes before the cell underwent interphase, before it prepared to divide. Okay, so the same type of chromosomes that we find, type, not number, guys, type. The same type of chromosomes that we had in the original mother cell before it underwent DNA replication during interphase is the same type of chromosomes that we are going to find in the daughter cells at the end of meiosis 2. Okay, so at the end of meiosis 2, we are going to have 4 daughter cells. These 4 daughter cells are haploid, and these 4 daughter cells are genetically different from each other because of crossing over that happened during prophase 1. Okay, so that is an overview of meiosis 1 and meiosis 2. Okay, guys, we have reached the conclusion of lesson 2 on our video series on meiosis. I hope this was a very beneficial lesson for you as it was for me. I do not have assessment questions for now for this lesson. The assessment questions I am only going to issue, and we are going to work them out at the end of lesson 3 on the following lesson. Remember, we have not talked about crossing over, random arrangement of chromosomes, genetic variation. So I will only be assessing you on the process of meiosis at the end of lesson 3. So if you want to check out the assessment questions, exam type questions for meiosis, I have a few at the end of the following lesson. Okay. Please do me a small favor. It's a small favor for you, but it means the world to me. Please react to my video, comment any contribution that you would like to make or any confusion that you might have. I will surely read all the comments and do my best to respond to all the comments. Subscribe to the channel so that the lessons or my channel will not be lost on the YouTube algorithm, and then share with friends, share with your teacher so that they might also benefit from the lessons. Okay. I appreciate you watching. The next lesson will be the lesson on genetic variation, where we will be discussing how meiosis contributes towards genetic variation. Goodbye.